This video will explore the idea of coherence in science instruction. The first half of the video shows the different types of coherence, emphasizing conceptual coherence. The second half illustrates how connections among concepts can be made and how these connected concepts are linked to the big idea. What does coherence in science instruction mean? Instructional coherence can have several different meanings. Coherence can mean the alignment of components of instruction, that is, ensuring that the nature of the assessment reflects the approach to content and pedagogy. Coherence can occur across grade levels, building conceptual understanding from one grade to the next. Coherence can occur by making connections among the different disciplines. And content coherence occurs when concepts are presented to show that they are interconnected and build upon each other. This video will be focusing on content coherence using concepts from the genetics unit from Foundation Science Biology. Every science discipline has overarching principles or big ideas derived from explanations for a range of natural phenomena. One big idea in biology is that all the genetic information required by organisms is contained in DNA. This big idea in biology is comprised of three components, which include the molecular basis of traits, inheritance of traits, and variation. Each component of the big idea is comprised of specific concepts. For example, understanding how genes are responsible for traits involves knowing that genetic information is encoded in the arrangement of DNA nucleotides, that genes encode proteins that are responsible for traits, and that transcription and translation facilitate the transfer of information from DNA to protein. Understanding how traits are inherited involves knowing that chromosomes pass information for traits from one generation to the next, that chromosomes are made of DNA and protein, and that genes are located on chromosomal DNA. And here we have the promoter and the coding region of the gene. Understanding how variation occurs involves knowing that most human cells have two copies of each chromosome and therefore two copies of each gene, or two alleles, one from the mother and one from the father. It also involves knowing that alleles can encode identical proteins or variants of a protein, and that variation in a trait can result from the expression of a variant protein. For example, one variant of a given protein may encode brown eyes, whereas another variant may encode hazel eyes. The big idea and its components give students a context for organizing concepts. This approach applies to any discipline and any big idea in that discipline. This kind of hierarchy is necessary but not sufficient in acquiring a deeper understanding of the big idea. What else is needed to achieve this? Too often, students are presented with concepts as disconnected facts. These concepts may be memorized, but if they are not developed sequentially in relation to other concepts and in the context of bigger ideas, they may not be understood or retained. When the time comes for students to explain what they have learned, their learning is actually just an accumulated jumble of facts to be regurgitated on an exam and then quickly forgotten. Deeper learning occurs when students encounter the concepts in a connected, sequential way. You would have an initial concept being introduced, and this concept here is about the structure of DNA as it relates to its capacity for encoding information. A second concept or fact is then connected to the first concept. Here, the second concept will be more deeply understood by making the connection to the concept about the nature of the genetic code and how information is stored as triplet codons. The next concept, chromosomes are made of DNA and protein, describes the structure of a chromosome and builds on the previous concept related to the structure of DNA. The next concept, genes are located on chromosomal DNA, builds on the structure and function of DNA. This concept, along with most cells have two alleles for each gene, and alleles can encode identical proteins or variants of a protein, are anchored in the previous concepts relating to the structure and function of DNA, genes, and chromosomes, and prepare students to explore how variation of traits occurs at the molecular level. Coherence lies not just in the sequencing of concepts, but also, and perhaps more importantly, in the many connections that can be made explicit among the concepts. Introducing concepts in this way demonstrates how the components of the big idea are related and how the web of concepts and components can lead to a greater understanding of the big idea. There is also more than one way to sequence the introduction of concepts. A learning pathway may start with the concept that variation in a trait can result from the expression of a variant protein, and then bring in additional concepts and connections over time. Although the order of introducing the concepts may differ, the connections among the concepts are similar regardless of the sequence. While there are different ways to sequence the introduction of concepts, students can only achieve a deeper understanding of the big ideas and related concepts by making the conceptual connections.